Dear students, the next organelle that we are going to discuss is Golgi apparatus. Now we'll discuss first what is Golgi apparatus. This organelle in the cell, it is a complex protoplasmic structure which is made of smooth membranes which are called as cisternae, a network of tubules with vesicles and vacuoles, and this organelle, it is involved in membrane transformations, secretion and production of complex biomolecules. That means the organelle is made up of membranes which arrange themselves into cisternae, and out of these cisternae some tubules, vesicles and vacuoles, they originate. This apparatus, Golgi apparatus, was first discovered by Camillo Golgi, and for this work he got his Nobel Prize also. And the organelle is visible both by light and electron microscope. That means it is a significantly big organelle in a cell. In plant cells, the Golgi apparatus, they are present in many number. That means they are scattered in the cytoplasm and they are unconnected units. So where such kind of arrangement of Golgi apparatus that is present, we call them dictyosomes. These organelle, Golgi apparatus, we know that prokaryotes do not have organelles which are membrane bound. So Golgi apparatus is absent in them, but it is present in eukaryotic cells. In some of the eukaryotic cells, such as your mammalian RBCs, the sieve tube cells of plants, sperms of bryophytes and pteridophytes, in these cells the organelle is absent in eukaryotes also. And more Golgi apparatus is found in those cells which are involved in some secretory function. That means the glands, the cells which are present in glands, they will have nicely developed Golgi apparatus because they are the ones which have a role in secretion. Then the position of Golgi apparatus is different and it depends upon the kind of cell it is present. A cell may have one large Golgi apparatus or several small ones that I already discussed to you, that sometimes dictyosomes are present. That means the Golgi apparatus, many small units of Golgi apparatus, they are scattered in the cytoplasm. Dictyosomes are also present in invertebrates. In secretory and absorptive cells, these are present. The Golgi apparatus, it occurs between the space present in nucleus and plasma membrane. Then the cytoplasm around the Golgi apparatus, the immediate cytoplasm around the Golgi apparatus, no organelles, no other cell organelles are present in that. वहाँ पे cytoplasm खाली रहता है, कुछ और ऑर्गेनली प्रेजेंट नहीं होगी, और इस जगह को जो है वी कॉल इट एज जोन ऑफ एक्सक्लूजन. यहाँ पे हर चीज एक्सक्लूडेड है, मतलब बाहर है, यहाँ पे नथिंग वुड बी प्रेजेंट. Now we'll see the structure of Golgi apparatus. Golgi apparatus, its structure, it varies in form and size in different types of cells, and if we see by electron microscope, then the structure seems to be like this. This is the structure of Golgi apparatus. The major element which forms the Golgi apparatus are these cisternae. These are the cisternae. इनको cisternae बोलते हैं, and each cisternae that is 60 to 90 angstrom in its it means it encloses a lumen of 60 to 90 angstrom. It's the 60 to 90 angstrom thick, and usually between the cisternae, between one cisternae to the next cisternae, there is a space of around 100 to 300 angstroms. This space is 100 to 300 angstrom, and this space between here it is 60 to 70 angstrom. And here a cytoplasm like fluid is present. Actually this is the cytoplasm of Golgi body, but it has similar almost similar composition to cytoplasm, and some proteins are present. So this is present in between them. The cisternae may be sometimes straight, or most of the time they are curved होंगे. So there will be a convex and concave face of the Golgi body. This convex phase is the cis phase or the forming phase, as you see here. The cis phase or the forming phase, ये phase एंडोप्लाज्मिक रेटिकुलम की तरफ जो है Golgi बॉडी का होता है. वेसिकल्स आके यहाँ पे फ्यूज करती हैं Golgi बॉडी के साथ. वेसिकल्स विच कम फ्रॉम एंडोप्लाज्मिक रेटिकुलम, वो यहाँ पे फ्यूज करती हैं. देन दे ट्रेवल थ्रू दिस ल्यूमन, एंड ऑन द अदर साइड विच इज द मैच्योरिंग और ट्रांस फेज, द कॉन्केव फेज, यहाँ से ये वेसिकल्स जो है पिंच ऑफ करके देन जो मटेरियल है इसके अंदर जो है मॉडिफाई होके देन इट इज पिंच्ड ऑफ इन सिक्रीशन vesicles. So this phase is towards the plasma membrane and called as the maturing or trans phase. And there are many fenestrations on Golgi body, means इसमें openings होती हैं Golgi body में, and from the openings you see the tubules originating, the vesicles originating, the vacuoles originating. ये जो यहाँ पे branched structure है, this is called as the tubule, and tubule they connect the cisternae. ये जो tubules है वो cisternae को connect करते हैं. And more about tubules is that they have a diameter of 30 to 50 nanometer, and some of these tubules they end up in vesicles. यहाँ से vesicles जो है वो पिंच ऑफ होती हैं. ये वेसिकल्स हैं जो पिंच ऑफ हो रही हैं, और ये वैक्यूल्स हैं जो पिंच ऑफ हो रहे हैं. ये जो वेसिकल्स जो पिंच ऑफ होती हैं दे हैव ए डायमीटर ऑफ 20 to 80 nanometers, and they are attached to the tip of the tubule. Sometimes these vesicles are coated, so we call them as rough vesicles. Sometimes they have smooth walls, they are not coated with any macromolecule, so they are called as if they are coated we call them as rough vesicles, and if they are not coated we call them as smooth or non-coated vesicles. These vacuoles they arise only on the trans phase, and vacuoles will directly arise from the cisternae. These vacuoles may sometimes change to primary lysosomes also. You can read the detail of the structure of cisternae from this particular note which I am giving you here. This is the tubule, vesicles and Golgian vacuoles. Then we'll discuss the functions of Golgi apparatus. Golgi apparatus, it is involved in secretion of material, in synthesis of material like carbohydrates, synthesis of glycoproteins and glycolipids, formation of lysosomes, formation of new cell wall material, transformation of membranes, apoptosis and sulfation. First we'll discuss secretion. So what happens in secretion is that all the glandular cells, जो secretion वाले cells हैं, glands के cell हैं, they depend on Golgi apparatus for concentrating and packaging their products. अपने products को pack करने के लिए उसकी vesicles में या vacuoles में जो है material pack होके जाता है, and they are sent to other organelles or for exocytosis or for reverse pinocytosis. The forming phase, it receives transitional vesicles. The forming phase, which is the forming phase? This is the forming phase. You can see here some vesicles, they are coming from endoplasmic reticulum. तो ये transitional vesicles कहते हैं इनको. ये transitional vesicles जो है वो endoplasmic reticulum से आती हैं. गोल्जाई बॉडी के साथ में इसका फ्यूजन हो जाता है सिस फेज पे. मटीरियल जो इनके अंदर था that will enter the lumen of the Golgi apparatus, and here they with the help of coated vesicles and intercisternal connectives, this material will pass through the cisternae, और वहां पे पास होते होते this material is modified, और modify होके जो है they are budded off. दूसरे एंड पे जो ट्रांस फेज है उसके ऊपर जो है ये जो modified material है वो vesicles में pinch off हो जाता है, and it is sent to the places where it is destined to go. Now, this organelle is also involved in synthesizing many complex carbohydrates such as the pectic compounds, mucopolysaccharides, hyaluronic acid, chondroitin sulfate, hemicellulose, and some simple carbohydrates like sialic acid and galactose. These are formed in the Golgi complex. Then we have transformation of membranes. Membranes are transformed. That means they are converted from one type into other type, and this happens in plasma membrane. One type of membrane, example that of endoplasmic reticulum, that will change to the kind of membrane plasma membrane is. So that means here recycling or formation or transformation of the membranes that takes place. Sulfation of molecules that happens in the lumen of Golgi apparatus with the help of enzyme sulfotransferase. Here even proteins and lipids which are synthesized in rough endoplasmic reticulum and smooth endoplasmic reticulum respectively. These proteins and lipids are carried to Golgi apparatus, and here they get attached by a carbohydrate moiety. That means they will be changed to glycoprotein, and if carbohydrate attaches to lipids, it changes to glycolipids. So this formation of glycolipids and glycoproteins that happens in Golgi apparatus. Formation of lysosomes. Lysosomes, I have already discussed to you how lysosomes are formed. Their pre-enzymes are formed in endoplasmic reticulum, and from endoplasmic reticulum these enzymes are carried to Golgi apparatus, and in Golgi apparatus then what happens? The enzymes they are taken into a new covering or new vesicle, and now this particular structure, the vesicle containing the primary enzymes, they are called as the primary lysosomes. New cell wall material is formed when the cell plate is formed. That last year we already discussed in formation of the cell wall, when the cells divide. So when between the two divided cells a new wall has to be formed, then vesicles, the vesicles of Golgi apparatus, they come and lie together and their membranes fuse, and the new walls they are created. Formation of acrosome. Acrosome is present in the tip of animal sperms which digest the covering of the egg during fertilization. So it is synthesized by Golgi apparatus with the help of its vesicles. Vitellogenesis, that is the in oocytes of the animals, the yolk is synthesized. That also happens with the help of Golgi apparatus. So this is another organelle, sphaerosomes. These are micro bodies like your peroxisomes or like your lysosomes. These are present in plants, and these organelles are related to the synthesis and storage of lipids. these organelles have another particular peculiar feature that they are bound by single membrane है और इसको हाफ यूनिट मेम्ब्रेन बोलते हैं. हाफ यूनिट मेम्ब्रेन मतलब हम जो मेम्ब्रेन्स पढ़ते हैं उनमें बाई लेयर है, जैसे प्लाज्मा मेम्ब्रेन में बाई लेयर है, लेकिन बाई लेयर का हाफ मतलब जस्ट one phospholipid का one layer that is present around these organelles, and their phospholipid the polar heads they lie outward towards the cytosol and their tails they are towards the inner side. So half unit membrane covers these organelles. These organelles were discovered by Perner, and they are found only in plant cells. The membrane of these organelles is stabilized by protein oleosin. Oleosin protein ही ये हाफ यूनिट मेम्ब्रेन को स्टेबलाइज करता है, इसीलिए इस ऑर्गेनेली का नाम जो है ओलियोसोम भी है. 98% of a sphaerosome is lipid. That means they are totally dedicated to storage of lipid. Rest is the protein, which is just 2%. Some proteins are probably the enzymatic ones, and others, these enzymes, they take part in synthesis of lipids. When we color these with the Sudan dye, then they get dyed, because Sudan dyes are used to color the lipids. So whenever we color the cell with the Sudan dye, these organelles will surely pick up the dye. Sphaerosomes they also occur abundantly in endosperm cell of oil seeds, because they store lot of oil, lot of lipids there, and these are spherical and oval in shape. Their diameter is just 0.5 to 2.5 micron, and they contain hydrolytic enzymes, proteases, ribonucleases, phosphatases. Hence they are called as plant lysosomes. So this is all with this. Next lecture I'll come with other topics like mitochondria and chloroplast. Thank you.